earlier today and this is not something you have to do immediately, but it's something that may show up on the exam. Remember, I asked you to display graphically what somehow to summarize the functional significance or whatever of the speed ratio and mechanical advantage for the animals from one of the very first labs that we did, right? We did the 2 monkeys and the armadillo and the opossum and the cat and the bison and the horse and all of that sort of stuff. And most of you, what you did was you made a bar graph and then you had, all these different things for each species, and it's really difficult to interpret what the heck that means. That's what almost all of you did, and that's basically what most people would do, but there is a better way to do it. Without doing any additional computations or anything, the idea is to present the data in a way that makes a point, right? What most people provided was so busy and so confusing, it's hard to even look at and figure out what's going on. And in general, if a graph requires a lot of work to try and figure it out, it's not doing its job. So I looked at the data and I said, okay, we have complete data for like 5 or 6 species, and then there was incomplete data for 2 species, so I just threw out the 2 species for which there was incomplete data. And then because speed ratio and mechanical advantage are simply the inverse of each other, there's really no new information there. So what I did, on this axis, I plotted the speed ratio of the femur, which is essentially going to tell you how fast the animal is. And on this axis, I planted the speed ratio of the humor, or of the ulna, right, which tells you how fast the front appendage is. Now, there are 2 ways to look at this. You can look at it from the perspective of isometry. So isometry is where they start at the origin, and then you just have a line that goes straight up like that. So if the speed ratio in one is equal to the speed ratio of the other, they're going to fall exactly on that line. But we know that mammals, with only a couple of exceptions, have hind limb dominance. So what I did instead was I took all of these points that were there in the data set, and I figured out sort of what the natural relationship was. Okay? And when you do that, you get a line that goes something like that. So it's basically the same idea, but it shows the, it takes into account the fact that we have hind limb dominance. So now this becomes our new sort of line of isometry. So anything that is below that line is going to have lower than expected speed ratio for the ulna. They're going to have slower front appendages. Anything that is above that line is going to be faster. And when you do this, you turn out, at least for the data from the one student that I looked at, that these were the monkeys up here. And this was everybody else. So these are slow animals and fast animals, right? And these are strong animals down here, or powerful animals, and quick animals up here. And there you can see this concept, these things right here, these arrows represent what are called, not derivatives, residuals. So these are the residuals. So if you're below the line, you have a negative residual. If you're above the line, you have a positive residual. That's your distance from that line of isometry. So the monkeys had positive residuals, everybody else had negative residuals. So what that tells you is that the monkeys, right, are using their hands quickly, right, whether it's to grab a branch or to grab something, right. Everybody else is emphasizing power rather than speed. So that is one alternative way to think about those data. And graphically, it's much more pleasing. It's not as noisy, it's not as busy. You can look at it and say, yeah, that makes sense, because now we're looking at everything in one very cohesive sort of way. Okay, so you don't need to worry about that. You've all earned the points for doing that graph if you turned it in. The other thing that is still outstanding was from the locomotion data. The part on humans, we have the human data, that's for us. The other thing that we need is to get to the point where I can give you some footprint data, and then you tell me how fast that person was going. Okay? So what I would do is I will give you the length of the footprint, right, how big the footprint was, and then I would give you what the stride length was, so the distance from there to there. And based on that number and that number, you can tell me how fast that person was going. Right? And if you look at the PowerPoint, right, all the information that you need is there. So we will go over that in lab on Thursday. Okay? We will not make that, well, no, let's make it due before the exam, because there will be a question on the exam pertaining to that. So let's make sure we know how to do that on Thursday. We'll go over it on Thursday. You can turn the assignment in on Thursday, and then you'll be have that off your plate, and you don't have to worry about it before the exam. Sound like a plan? Sounds like an awesome plan. All right. The next thing that you need to do, I don't know if it's reasonable to ask you to have that done before the exam or not, but has everybody downloaded the video, been able to download the video? So it's there on YouTube. Everybody know how to get a video off of YouTube? There's a software program called, I think it's called 4K Video Downloader. It's freeware, okay? They don't put any weird little things on the video clip. You can grab as long a video as you want. If it's on YouTube, you can get it. All right? So it doesn't take a whole lot of room on your hard drive. It's a pretty small program, but it's really effective. You open the program, you have YouTube open, you cut and paste or cut and copy the link from YouTube, paste it into 4K video downloader, and then it automatically downloads it. And you can specify how you want it downloaded. Okay, what file format you want. Okay, there's another program called, I think it's called File Converter Lite. And that program will allow you to convert any file, any video or audio file from one format to another. So if the video file is an AVI file, you can convert it to an MPEG or whatever you want. You can specify what the pixel dimensions are, whether it's going to be 4K, 5K, whether you want it 260 by 480 or something, whatever the dimensions are, you can specify that. You can't increase the resolution of something, but you can decrease the resolution of something. Okay? If you want to go to YouTube and steal the music without the video, right, you can download your music video, strip off the video part, and save the audio as an MP3, and then put it on your phone or wherever you put your music. Okay? Same sort of thing. So it's two very handy programs to have. So you can use that to grab the video, you can use that to strip off the audio, right? And then you can simply import it to IrfanView. You can trim it down a little bit in Movie Maker, get rid of all the extraneous footage that you don't want. How long is the video now? I think it's
10 minutes, I think.
10 minutes. So there's a lot of antelope stuff on there. You don't need the pronghorn, all of the pronghorn antelope stuff. You just need a little bit of it. Probably half that video is pronghorn. Okay, male pronghorn running, galloping, walking, ambling. The pronghorns do a whole series of different gaits. There is that African antelope that's in there, I think. Did I include that African antelope in there? He stands in one place, but you notice what he does with his feet, right? So he's stomping his feet. What the hell is that all about? Any other animals do that? Yeah, lots of animals do that, including pronghorns. Except pronghorns do it all at the same time, right? Pronghorns have this really bizarre gait where they'll just stand there and suddenly they're airborne and then they land exactly where they started from. It's called pronking. It's kind of cool. All right, we're all good then. Ortho-J, you need that. The locomotion data, the human stuff, we're going to work on that on Thursday. And then this stuff, the gait analysis. So I think we'll make that due after the exam. So we don't want to take time away from your prep for the exam. Very good. Let's get started then on the material for today. What we've done, if you think about it, is this, and there are a couple of points that I want to make. Let's look at a very sort of abbreviated phylogeny for the vertebrates, and we've kind of already done that. We have fish, right, and then down here we have amphibians. And then very early from the amphibian radiation, we have the reptiles. Then coming off very late from the reptiles are the birds. And very early from the reptiles are the mammals. The mammal-like reptiles are there, right, at the end of the Permian. Now, they're not mammals, they are reptiles, right? But they do have a lot of mammal-like attributes. So they have, there are some interesting differences in the skulls of these guys, okay? And we'll talk more about those in just a few minutes. But we've made a couple of points. The first thing we noted was sebaceous glands, so skin glands. Everybody has skin glands until you get to reptiles and birds. So here the glands are lost. So the skin glands are lost. Birds still have one. These guys have a two-chambered heart. So initially everybody has a two-chambered heart. But the amphibians have a new invention. They have a three-chambered heart. Then, somewhere in here, we get a 3.5 chambered heart. Here, we get a 4-chambered heart. Here, we get a 4-chambered heart. It's easy to get the 4-chambered heart. All you have to do is extend that septum that goes down the middle of the ventricle. But birds do it, mammals do it. There are a couple of weird reptiles that do it, the branded lizards and the crocodilians. Crocodilians are archosaurs and the probable ancestors for the birds. I know everybody says that dinosaurs are ancestors to birds, but don't forget dinosaurs are also archosaurs. Okay? So there's the 4-chambered heart. Reptiles, for the most part, still have the 3.5-chambered heart. What else is different amongst those animals? So there's something going on with the heart, with the circulatory system, with the skin glands, Okay, are there other attributes? Yeah, there's a whole bunch of other attributes. And let's get into some of those now as we start talking about that first radiation of the reptiles. So there is sort of this interesting thing that takes place. Let's define what a reptile is. Reptiles possess an amniotic cleidoic egg. What that means is around the egg there's an amniotic layer. And then there's this hard layer around that, so it's the shelled egg. That has certain consequences, okay? So we have this, the amniotic sac. You guys have an amniotic sac. We have the chorion. Well, you guys have a chorion membrane. The allantois, we've got that. A yolk sac, well, we do that, but we kind of replace it with a placenta. But reptiles also have a placenta, as do birds. Okay? Their placentas are fundamentally different, right? The placentas in reptiles and monotremes are essentially identical. The placentas in eutherian mammals are different, but there are some marsupials that have placentas that look very much like ours. Okay? So there's this weird thing that goes on with the marsupials. So cleidoic eggs are leathery eggs that are usually pervious to water, but sometimes they are impervious to water. If you find a turtle egg or a snake egg or a lizard egg and you just put it in a warm place hoping that it's going to develop, what will end up happening is it'll dry out and shrivel up. Okay? So reptiles are very smart about where they deposit their eggs. They deposit them in places where you can get water exchange with the soil. Sometimes the egg becomes calcified and impervious to water. In any event, what that means is that you have to have internal fertilization. So reptiles, without exception, have internal fertilization. That's something that only some amphibians do. Clearly, some fish have internal fertilization, right? But all reptiles have to have some sort of internal fertilization. And what that means is you have to have some kind of an intromittent organ. So the male has to have some device that he can use to insert into the cloaca of the female to transfer sperm. And they do it in all sorts of cool and interesting ways. Now, before we go too much farther, I want to talk about two people. I want to talk about this guy. This is Edward Drinker Cope. He's been in the news lately. He's been dead since the late 1800s, but he's been in the news lately. Does anybody know why? Something about a professional journal? Yeah.
Who Copia is named after.
Yeah, the journal Copia is named after him, or was named after him. It is no longer called Copia.
Was it race? Or was it sexual harassment?
Oh, no. He was a racist.
Yeah.
But let's keep that in context. Benjamin Franklin was a racist. Thomas Jefferson was a racist. Thomas Jefferson had slaves. Edward Drinker Cope never owned slaves. Okay? So that brings up an important point. Okay? At the time, and I'm not justifying it, but at the time, right, the races were not considered to be equal. The Christians didn't consider the races to be equal. How do we know that? The Jews didn't consider the races to be equal. How do you know that? Read the Old Testament. In the Old Testament, it talks about who you can have as a slave, what you can sell your slaves for. You can keep slaves from neighboring countries. You cannot keep slaves from your own country. So they recognized themselves as being superior to people that lived in other countries. Okay? How did we justify slavery in this country? How did we justify the slaughter of the North American Indian? What term did we use to describe the Indians?
Savages.
Savages. In other words, they weren't human, they were animals. Okay? How did we deal with the slave issue. How did we refer to them? They were livestock, okay? They were treated as livestock. They were not considered to be humans. That's how we justified keeping them as slaves. So how is it possible? Well, that's the Christian way. How do I know? How can I make a statement like that? What basis does that have in Christianity? Well, what's the underlying philosophy for Christianity? Which Greek philosopher forms the basis for modern Christianity? Well, what's the highest form of love? Platonic love, right? Platonic. Okay, so now, who's the philosopher that has the ideas that basically formed the foundation for Christianity? Plato. Okay, and did Plato have slaves? Yes, he had lots of slaves. Was Plato a Christian? No, he wasn't. Okay, but what did Plato do? Plato believed that the world, the universe, the cosmos could be explained with numbers. And when he discovered that there were irrational numbers, like the square root of 2, he couldn't understand it. So what he decided was that that information was too dangerous for the common man. So they concealed that information. Some knowledge was too dangerous and should only be kept by the people like Plato. It was essentially a cult of mysticism. And one of the things that Plato did was he had slaves. His justification for having slaves was that his slaves weren't intelligent enough to understand the numbers and the systems that he was working with. They didn't have souls. That's where the idea of separation of mind and body comes from, which is what Christianity is all about. Substitute soul for body, and there you have it. Right? So what we're talking about is separation of soul / separation of mind from the body. When you die, if you're Christian, when you die, your soul goes either to heaven or to hell, right? That's what they taught us when I was in parochial school. Okay? So that's where that comes from. Thomas Jefferson, everybody back in those days was essentially a racist because slaves, black people, were clearly not human. They were animals. They were livestock. So, an interesting thing about Edward Rinker Cope is that he was a Quaker. Anybody know who the Quakers are? They're not much in the news anymore. Name one president who was a Quaker. Before your time, but not that long before your time. Let's see, Jimmy Carter is, what is he? Is he a Baptist or a Methodist? I think he's a Baptist. Bush. What were the Bushes? Not Baptists. They must have been Methodists or something. Clinton? Who knows? Episcopalian, I think. Kennedy? Catholic. Who came before Kennedy? Eisenhower. What was Eisenhower? Who knows? Nobody asked in those days. Kennedy was a problem when he ran for office because he was Catholic. And the American population was concerned that the Pope would run the country if he became president. Okay? Richard Milhouse Nixon was a Quaker. Okay? One of 3 presidents to be impeached. Well, actually, Nixon was never impeached, right? He resigned. When he saw that the vote was coming, he resigned. Back in the days when people had honor, okay? Back when you didn't get a blowjob in the Oval Office, or you didn't, sell your soul to the Russians or whatever. Okay, yeah.
I would just like to point out a bit of irony about the coronavirus treatments that Trump is receiving.
this, whatever the monoclonal whatever that they have them on is
Yeah.
derived from fetal stem cells. That's kind of funny.
It is.
This
It's,
whole pro-life base,
I know, but
but
well, let's be honest though. Let's remember that there is not a politician, and I don't care which party,
no.
there's not a politician out there that isn't giving you a sales job.
oh no.
Okay, it's all a sales job, and the truth has got nothing at all to do with it. Okay, just keep that in mind. So it's, at any rate, why do I bring up the point that Richard Milhouse Nixon was a Quaker? Well, because it turns out that Nixon did an awful lot of stuff in favor of the environment. Okay, so a lot of government policies that are currently being dismantled were originally put in place by Richard Nixon. So as evil and as nasty and as rotten as Nixon was, he did some positive things. Okay? The reason he did those things is because he was a Quaker, and the Quakers are naturalists. They are people that have a keen interest in the natural world, and that was true for Edward Drinker Cope as well. Edward Drinker Cope, his father wanted Cope to become a gentleman farmer, and if he wasn't going to become a gentleman farmer, he wanted him to become a man of the cloth in the Quaker religion. Okay? But he came from a family of money. This guy is Othniel Charles Marsh. He was the nephew of George Peabody, the same Peabody that you see across the river from the Arch in St. Louis, Peabody Coal, right? So his uncle was an industrialist back in the 1800s, filthy rich, donated lots of money to all sorts of causes. He was a frat boy. He didn't graduate from high school until he was well into his 20s. He wasn't a very good student because he liked the ladies and he liked his booze. Okay, he wanted a life of leisure. And the other thing that O.C. Marsh did, he started a scientific journal called American Men of Science. Edward Drinker Cope also started a journal called The American Naturalist, which is still, it is today the premier theoretical ecology journal on the planet. Okay, so these two men began their careers in close proximity and as friends, and they grew apart and they ended up being bitter rivals. There are lots and lots and lots of YouTube videos and books which detail the lives of these two men. It's really an interesting read. But obviously the journal American Men of Science didn't last very long. It went extinct. At the end of his career, he had published 600 scientific papers all about dinosaurs, and not mostly about dinosaurs, but lots and lots and lots of mammal material as well. He wasn't a very good student, and when he was finishing up at Yale, he convinced his uncle to make a large donation to Yale University. And when they made that donation to Yale University, it was for the purpose of the Museum of Vertebrate Paleontology at Yale. So George Peabody contributed all the money necessary to start the Museum of Vertebrate Paleontology at Yale. One of the stipulations in that donation was that Othniel Charles Marsh be hired as the curator of paleontology at Yale for life. And they did hire him. And his salary was $1 a year. Okay? So he was basically working for free, but that's okay because he was rich, right? And he had a prestigious job at a major university, so he got what he wanted. At any rate, he published 600 papers in his professional career. Edward Drinker Cope, on the other hand, published 1,600 papers. A record that was only surpassed a few years ago by the guy who wrote my comparative anatomy textbook, a guy named Hobart Smith, who at the age of 93 published his 1,601st paper, finally breaking the record that was set by Edward Drinker Cope back in the 1800s. Edward Drinker Cope didn't get very old. He only got into his 50s before he died. So, at any rate, these two guys were bitter rivals. They hated each other. They started off as friends. There are some interesting stories. I invite you to look at some of those. I'll post some of those little video clips about these two guys online. You should take some time and just view those for your own amusement. But they are the foundation of basically of herpetology and certainly of paleontology. They are responsible for most of what we know about dinosaurs today. Okay? Just as a side note, they ended their lives, they ended their careers and their lives. Both of them were broke because they squandered their money fighting each other in these bitter battles. Edward Drinker Cope was one of the founders of the U.S. Geological Survey and ended up losing that position and involved in a lawsuit with O.C. Marsh and then was countersued. And both men ended up losing their collections and essentially being broke and ruined. But the final coup de grace was, and this is sort of interesting, at the time, the idea was that a bigger brain meant you were smarter. So that people viewed brain size as being related to intelligence. Well, it's easy to see where we would get that idea because humans have big brains and we view ourselves as being really smart. Okay? That is also the idea that led to Morton's paper, which at the time, quote unquote, proved that whites were superior to Asians and Asians were superior to blacks. Okay? At any rate, Edward Drinker Cope, before he died, in his will specified that his skeleton be preserved, cleaned, deposited in a museum, and he recognized that there was no type specimen for Homo sapiens. Every species has a type specimen. And he knew that there was no type specimen for Homo sapiens. So he wanted his skeleton to be used to define the species Homo sapiens, and he wanted his brain volume measured and then compared to Cope's, compared to Marsh's brain size when Marsh died, to prove once and for all that he was smarter than Marsh. Okay? So it wasn't until about 15 years ago that somebody tried finally to complete that last part of Cope's will, and that is define him as the type species. So what's his name? Backer, the guy who wrote the Dinosaur Heresies. A student under John Ostrom. Ostrom is the guy who described Deinonychus. Ostrom is the guy who described or came up with the idea that the modern idea that birds are dinosaurs, right? One of his students, Robert Backer, was a student of John Ostrom. John Ostrom was the curator of vertebrate paleontology at Yale, so occupying the same seat that O.C. Marsh had. At any rate, Backer and another fellow get Cope's skeleton. They write a paper describing him as the type specimen for Homo sapiens, submit the paper, right, to get it published, and then take Cope's skull on a world tour. They went to every continent, to every major museum, to every major university, showing off Cope's skull, trying to push the idea that Cope was the type specimen for humans. And ultimately, the Swedish Academy said no, Cope can't be the type specimen because he didn't have all his teeth. He had false teeth. And the type specimen has to be perfect. So his skull was not perfect, and for that reason, he was rejected as the type specimen. To this day, there is no type specimen for Homo sapiens. So what I'm telling you, if you want to be famous, and your skeleton and your skull and your teeth in particular are perfect at this point, hell, die young, put it in your will, you want to be the type specimen, convince your buddies to process your body appropriately, write the paper, you'll be immortal. You'll be sitting in a museum somewhere, researchers from all over the globe will come and look at your skull, it'll be great. Okay. All right. More about those two guys another time. So let's look at what happens in the development of the reptilian egg, okay, or the reptilian embryo. So there are two sacs that are important. The first is this allantois, right? So there's the allantoic membrane. Here's the gut, and then here's the yolk sac. Well, we have a yolk sac as well, except that it's really minimal, and this actually in mammals becomes the umbilical cord. That then would be the placenta. So we have a placenta, which is referred to a little bit differently. These guys have a placenta that is the choriovidulan placenta. We, mammals, have a placenta that forms through the allantois. We have a chorioallantoic placenta. There are two different types of placentas, chorioallantoic and choriovidulan. Vidulan, as in these are your vitals. Okay? All right. What's the difference? The difference between those placentas is the amount of innervation that you get with the uterine wall. So, in a reptilian placenta, it looks like this. Here's the uterus. Well, okay. They don't have a formal uterus, but there's the tissue, and there's what the embryo connection looks like. In mammals, it looks like this. What's the difference?
There's more surface area in mammals.
More surface area in mammals. Okay? So the exchange of oxygen, of gases, of nutrients across that placental interface is much more extensive in mammals than it is in reptiles. Well, it turns out that this is the way placental mammals do it. Placental mammals plus numbats plus one other group, and I've forgotten what the other group is, okay? But there's one group, at least one group of marsupial that does it this way. This is the way marsupials and reptiles do it. And monotremes. What are monotremes?
Are those the mammals that lay eggs, right?
Yeah, duck-billed platypus and the echidna. So the duck-billed platypus and the echidna lay eggs. And they have a placenta just like a reptile does. So that interface, just like the reptile does. So notice what happens, right? The yolk sac, the yolk gets used up in this egg, right? What happens in the allantoic cavity? Well, there's the amniotic sac, there's the allantois, right? So that allantoic cavity is basically this protective layer that goes around there. There's that amniotic sac, right? That's this protective layer that goes around the embryo. What's happening is that all the waste material is going into the allantois. The yolk sac, that contains the food, that's where the nutrition is coming into the animal. The key is that this whole embryo is totally encased, right, by this chorionic layer on the outside. It's this egg which is totally protected, okay, so the little embryo is developing in this very protected kind of environment. What that means is that these animals have greater protection against the elements. So when the egg is produced, when the egg is laid, the female can walk away from it and is free to do whatever. That egg, that embryo, can now complete development in a protected place without as much risk of predation, without as much risk of desiccation and all those sorts of things. So it's an interesting strategy on the part of the female. There are counterexamples, right? Are there any weird things that reptiles do with their eggs? Sure. Some reptiles, like red-tailed boas, so boa constrictors, what do they do with their eggs? Where do the eggs hatch? Where do they lay their eggs? They don't lay their eggs. They keep their eggs inside the body, okay? The eggs hatch inside the body, and they then give live birth. Okay, so why would they do that? So you have two strategies. You can deposit the egg somewhere, walk away from it, or you can keep it with you, keep it in your body, and then at the right time deposit all these things at once. Give live birth. What's the cost? What's the advantage?
Well, the advantage of giving live birth is like you don't have to worry about whether or not something's gonna come and get the eggs, I guess.
Yeah, you don't have to worry about the raccoon getting them.
Yeah.
Okay, you don't have to worry about any of that sort of stuff. So the probability of completing development is probably a little bit higher unless you get eaten. Okay, which for a big snake is not likely. Okay, any other advantages to keeping these things inside your body?
Nutrition.
Well, all the nutrition is inside the egg. So the first thing that happens in a reptile, if you talk about parental care, the parental care that a reptile provides is the yolk. What do you accomplish by keeping the eggs inside of your body, aside from keeping it away from the possible predators, nest predators? What else are you accomplishing?
Aren't you like regulating their temperature?
Yes, because you, as an adult, as an individual, are able to carefully regulate your body temperature, you're also regulating the temperature of your eggs. You're able to keep those eggs at a higher temperature. Why is that advantageous? Because development is going to be faster at higher temperatures, which means you can produce the eggs quicker and then reproduce again quicker. If you lay the eggs in a nest somewhere, the rate of development is likely going to be slower. So by keeping them inside your body, you can bring them to fruition faster than if you put them in a nest somewhere.
Don't some reptiles like what heat the eggs are at determines the sex?
Yes, that's true in turtles, and that's true in crocodilians and a number of other reptiles as well. The interesting thing, they have temperature dependent, so temperature dependent sex determination. Okay? In turtles, hot nests produces females. In crocs, hot nests produce males. Why? Well, what's different about a turtle and a crocodile? Obvious, the obvious difference between the two. Pardon?
The shell.
The shell. So if you have a shell that encases your body, imagine human females had a shell that encased their bodies. What would happen when they got pregnant?
Crush their inside.
It'd be very uncomfortable. There'd be no room for anything. In other words, it would limit the size of the baby. So if you're a turtle, as a female, you're limited on the volume of eggs that you can produce. You can either produce a lot of little tiny eggs or you can produce a few big ones, but you're limited. Why would that then lead you, when you have a hot nest, to produce females. Well, what is a hot nest going to do?
Increase growth.
Increase the rate of growth. So metabolism goes up, growth is faster, so if you put a nest in a hot place, the eggs become females, they go really fast, so the females end up being bigger than the males. So you have reverse size dimorphism. Why do you want a bigger female? Because a bigger female has more gut volume, which means she can produce more eggs. So she can have greater fitness if she's bigger. The male, it doesn't make any difference because sperm are small. Why is it the opposite in crocodilians? In crocodilians, the hot nest produces males. Well, the female doesn't have a shell, so if she's going to carry a lot of eggs, that's okay. She just gets thicker and thicker and thicker. Why is it important for males to be bigger? What do crocodilians do?
Aren't they like I guess, I want to say polyamorous, but that's not the real term.
Well, they're polygynous.
That's what it.
Yeah. So what's the advantage to being big as a male?
More dominant,
Yeah, which means what?
better chance of actually reproducing.
Yeah, more access to females.
More,
Okay.
yeah, it's more competitive.
That's right. So if you're bigger, if you're in a hot nest and you're bigger, that means you're more likely to reproduce. So it's a reproductive strategy. What do the animals do? Because the turtles and the crocs select the nests. They know where they're putting the nest. They know, is this going to be hot or is this going to be cold? So they're adjusting it on the basis of what their perceived sex ratio of the population is. If there's a paucity of females, they'll produce females. If there's a paucity of males, they'll produce males. And they know exactly how to do it. Okay, we'll stop there and pick up with this probably after the exam.